Um, Good evening and welcome to everyone for the August WAS mini conference. We're over halfway through a year now and we've had these monthly talks tonight. It's all about bee health. A couple of comments. One is that our first speaker, Mr. Dave Wick, who will address you fairly soon, he is also the sponsor of tonight's webinar. Normally, and let me make this very clear. I asked Dave to talk and address WAS because of unique things he's done through his analytical services to bees and beekeepers. And so once he did, he generously volunteered that he would pay for the license fee for the presentation tonight. So it's not like, so I assure you, Dave didn't pay me so that Dave could talk to you. I asked him, and then he generously said he would defray the cost. So I'll make that clear. For those, I think everyone probably that is logging in has probably attended one or more of these before, but you are in a webinar format, everyone except for the speakers. And so there's a chat and a Q&A on the bottom line. And if you want to say howdy to somebody that you see a name and they say, oh, I know that person and so on, or you have a question for Chris, and Chris here is our astrophysicist IT guy that troubleshoots and keeps things running. So if you're having trouble with your Zoom connection, or you have something, or how do I do this, or can I do this, ask Chris through chat. So use chat for kind of, the howdies and generic comments and to colleagues and so on. Use the Q&A for specific questions that will be addressed to the speaker. The format is each speaker has 30 minutes to talk. And then at the end of the 30 minutes, I will moderate questions and I will take those questions from the Q&A that you folks generate. So kind of rules of the game here, what we're doing. So our first speaker is Dave Wick. He has a very unique laboratory that has been set up here in Florence, Montana. It basically has transitioned some unique technologies, analytical technologies from the U.S. Army into the private sector. And so it's a tech transfer, one that has been going on for many years now, and he continues to build the capabilities of his laboratory. Our second speaker, who is right now probably grabbing a quick dinner because he just got through addressing a bunch of people on the East Coast, is Dr. Dewey Caron. And so roughly about top of the hour, Dewey will come in and talk about the Honeybee Healthy Coalition and the work he does as a member of that coalition. So I think with that, Dave, I will turn it over to you. And you're about 3-4 minutes after, so you got about 34 minutes, about 5 minutes before. If you don't indicate that you are about ready to wrap up, I will throw in a comment and say about time to wrap it up, Dave. So it's my pleasure to introduce you to Dave Wick, and I will let you, since you're wearing two hats for two companies, describe your companies.
Okay, well, first off, I'm going to bring over my PowerPoint and get it running. And got to do this. Okay, there's my company, BVS Inc. I'm out of Stevensville.
You're not up yet.
I'm not up. Oh, I got to share the screen, don't I?
Yep. Yeah, that's fine. Bring your PowerPoint up, then go to share screen.
Then do this, click that, then come over here and do this again.
There we go. Now you need to flip your screens again. There you go.
There. That's where I started. That's
there you
BVS.
go.
That was a hike to the top of St. Mary's Peak here in Ravalli County, and then looking west across what Lewis and Clark looked at. I've always loved that picture. Okay, so I am going to move this over to another screen. BVS was as Jerry said, brought into the honeybee industry a long time ago. I met Jerry at the University of Montana. We were in a business card exchange with the university and the professors, meeting the business community. And Jerry and I kicked it off pretty well. when we saw that we both had a connection at ECBC, Edgewood Biological Chemical Command, where Jerry worked with honeybees, and I was working with the point detection team and looking for viruses. And we changed cards, and sure enough, read in the local newspaper that Jerry was down in Florida looking at honeybees, and he was quoted as saying, "We think this is an unknown virus that's affecting the honeybees and this die-off." So I go, well, wait a second, unknown viruses. I picked up the phone, called Jerry, and it was, he goes, "Oh yeah, that virus guy from the Army. Oh yeah." So we went forward and we got some samples sent to the Army base, and next thing you know, we're getting samples, and we ended up presenting, Charles presented at the ABF meeting, and the beekeepers, they needed a test for viruses. So they made the decision, the state of Montana, the California beekeepers, Wyoming beekeepers, they pooled their money, and we bought the equipment, put it in a lab, started out next to Jerry up in Missoula, and ended up moving to Florence. And then a couple of years ago, I moved it off to Stevensville. And we have covered a lot of territory, but here is the technology. I'm briefly going to go over this. And the piece of equipment is this electrospray. It's a classifier. All the classifier does is move the air through these hoses back and forth to keep everything moving. And then we have a DMA, which is a differential mobility analyzer. And then it goes to a particle counter. And 5 minutes later, 10 minutes later, I get the results. This happens to be influenza A that we looked at. Okay, so this little thing right here, that's where I take the prepped sample. We prep the sample, we filter it, we concentrate it. I take the sample, I run it in there, we electrospray all the particles in the sample. When you do that, everything is charged. It's basically a mass charge ratio. We take that and we neutralize all of the gas, the charges in the gas. It moves to out the port into the differential mobility analyzer. And that center rod is a charged rod with different bins on it, basically of the different mass charge ratios. All the particles that come out of the electrospray come down this column, and they stop at the corresponding charge. Every particle is counted or charged, and then we count it. The mass charge ratio can be differentiated and calculated back into the size. And so basically we sort the viruses out by their size. This little box down here next to the computer is just simply a condensation particle counter. We condensate butanol on the small particles because the particles are smaller than the laser wavelength. The particles all go through the particle counter single file, and we just simply count each one that goes through for every bin from 7 nanometers all the way up to 289.3 nanometers. And we sort them out, and that's how we look and see what's in the sample. This is a chart that I made for Sacbrood virus. Most of this talk tonight's going to be on Sacbrood. There's a lot of viruses that we can find, we can look at. I've seen just about everything that there is to look at. And this is what a typical readout looks for. It's 32.2 nanometers. It's important, remember that size. That is a Sacbrood virus. We put Sacbrood in, we got Sacbrood out. This was from a beekeeper that we looked at. So remember this is what my data chart looks at. Down here in the bottom line, there's 7.37 nanometers, 289 nanometers, 8.2 nanometers, 9.14 nanometers. Nanometers are very small, and we'll come back to that. So confirmation by proteomics, mass spec proteomics, we go to the gene bank, we go to NCBI, we download the reference material for Sacbrood virus, which was one of the first sequenced viruses that we have. And that sample that we just looked at actually happens to be the only virus in this sample. That's why I use it. And there it is, and it's highlighted here. That's Sacbrood virus. And our confidence level over here is very high. That's what we have. That sample, I had a single peak right there. I have a single detection in proteomics, and then we ran a real-time PCR validation on it, and we ran several different viral tests. The only one that was positive was Sacbrood, and this is how the PCR validation looks, and this was done by an independent lab. The proteomics was done by the Army lab, and my confidence in Sacbrood virus in what we pick up, I would say is very high. So what is it related to? Here's a phylogenetic tree. They're relating, this was a 2004 put out by Judy Chen and her publication. And there you can see the viruses, Sacbrood being down here in the lower second one up, and it's related to the Kikugo virus, the deformed wing virus. Notice the relationship between that, but I don't pick up, and I haven't seen anybody talk about the Kikugo virus. I do pick up Kashmir bee virus. It's related to the acute bee paralysis virus. Black queen cell virus is up over here. And it's just an interesting relationship. I don't have anything on Judy's paper for a phylogenetic tree back in 2004 of the relationship to Sacbrood virus. Here are a bunch of the different viruses that we can look at. Again, Judy Chen was in on this. I think it was Judy Chen was on this. You can see the different phylogenetic trees and the relationship. And Sacbrood virus is down here at the bottom again. And it's off here related to La Jolla virus. I do pick that up occasionally. Lots of plant viruses, other viruses, slow bee paralysis virus, deformed wing virus, again, Varroa destructor virus, closely related to the deformed wing virus. Very interesting data. Sacbrood virus, I caught it classified at 32.2 nanometers in size, condition affecting the brood and the honeybee, resulting in larval death. They failed to pupate. Fluid it forms around the pupa in the sac, and it fills up with liquid and accumulates, and it forms a sac, and that's why the condition is named. After the larvae die, they change color from pearly white to a pale yellow, and shortly after the death, they dry out, forming a dark brown. You can easily see this in the comb. But on the other hand, the Sacbrood virus may also affect the adult bee. So here's what the comb looks like. We got this from the Bee Informed partnership. Rob Snyder posted these pictures, and they're great pictures. Good for Rob in putting them up. And there's the link if you want to go to look to them. The National Bee Diagnostic Center, May 2114. This presentation hasn't happened yet. I like that. It's the first time I've seen that. So back in from Bailey's work in '72, talks again, it accumulates in the brains of infected bees, especially of drones, without causing symptoms. Not causing symptoms. What are the symptoms? Well, they turn yellow in the pupa and they turn brown when they die. Oh wait, that's in the comb. They don't show symptoms in the honeybee. That's what's really interesting to me when we first started finding these, is that we can use the Sacbrood virus to determine how stressed out the bees are by using the Sacbrood virus to give us an indication. The more Sacbrood virus, the more your bees are stressed. Okay, so Sacbrood virus in the honeybees. This is a paper written in 2001. Rapid identification and phylogenetic analysis using RT-PCR. That's what I showed on that earlier screen. This is the common way that everybody looks at it. There's been no other places, but I wanted to bring this one up because they are speculating that the Sacbrood viruses are 28 nanometers in diameter. 28 nanometers in diameter versus my 32. They use a TEM, electron micrograph, and they put a bar down on it, and they get as close as they can to how that virus that shows up in the TEM relates to that bar. So 28, 29 nanometers, 30 nanometers. When I was in college, they all came out at 30 nanometers. One thing I wanted to spend some time talking about was the trade-off between sperm viability and the immune proteins and the how Sacbrood virus affects the drones, affects the queens, and moves back and forth in that. This was written by one of the authors, Jeffrey Pettis and Leonard Foster and David Tarpey out of Georgia. Leonard Foster's in the University of British Columbia, and when I met Jeff, he was the leader of the ARS team at Beltsville, and I think now he's has his own company named Pettis and Associates. And I don't know the other two people, probably should. So out of their abstract on this, you know, they talk about the bees and others will keep the sperm alive within their specialized storage organ for years, define the typical trade-off between lifespan and reproduction. However, whether honeybee queens experience a trade-off between reproduction and immunity is unknown. That's really interesting. But they're going to come in and they're going to talk about how the bee health, their immunity affects the sperm, how that sperm is staying alive, and there's a balancing act that the queen does between being immune and then keeping the sperm viability. One of the things that they found in this study was that the queens rated as failed by beekeepers had lower sperm viability in the queen. Fewer sperm, higher levels of Sacbrood virus and black queen cell virus. I don't see a lot of black queen cell, but I do see it. When I am looking at it, I do see Sacbrood virus. And I track the Sacbrood virus coming and going out of beehives, out of the worker bees. I've traced them all the way from queens, worker bees, the nurse bees, the guard bees, and you know, and it changes over the year. I've got a chart that'll bring that out. Both the viral and the sperm viability data were highly variable. And I believe that they were probably all over the place on trying to track their viral load and the sperm viability. Oh my hat's off to these guys. However, they did say that failed queens tended to have higher viral loads as a lower sperm and as well as a lower sperm viability, which is consistent with a reproduction immunity trade-off. So when that first slide I showed, when I am doing testing, screening for your bees, you send them in to me, and everybody goes, yeah, we got the wonderful Sacbrood virus that you're always telling is there. Pay attention to it, because when you go back and start looking at your brood patterns that the queen's laying, and you're blaming that spotty brood pattern on something else, it may be back to the viability of the sperm in the queen. She's laying sterile eggs. Very interesting thing, something to pay attention to and keep track of on your beehives, on your bee yards. Sacbrood has a high mutation rate, which contributes to the vast diversity of strains circulating in both Apis ceranae and Apis mellifera. I asked Jerry earlier how many strains of Sacbrood virus, and this goes back to 2019. These guys found 52 strains of Sacbrood virus. Wow. How many strains are being looked for by PCR? PCR is very specific. You can create some crossovers, and I think there's 3 or 4 crossovers that you can pick out of different strains of virus, and you can get a detection for these different strains, a combination of 4 or 5 strains, maybe. 52 strains. When I pick up Sacbrood virus and you send it off to somebody else and they go, well, there's no Sacbrood virus in here. My reliability that I've got a 32 nanometer peak in my sample, it came from honeybees, it's probably Sacbrood. If they come up with a negative, well, PCR also has a 30% false negative and a 30% false positive. That's difficult to deal with, but part of their false negative, that's if they know and the primers are correct for that strain that they're looking for. If they don't have the primer for the correct strain, they're blind. They don't see it at all. And that's too bad. If I pick up Sacbrood virus, that's something to pay attention to. And I also quantify this by counting the virions in the sample. There's all sorts of stuff, the phylogenetic tree. One of the important things that they bring out, there's 2 distinct lineages, and based on the geographic distribution, and that is that the Sacbrood virus affects Apis mellifera and Apis ceranae, 2 different sets. That's really significant. And our virus here in the US are mostly related to the United Kingdom and Sweden strains of Sacbrood virus. Wonder where that came from. And Sacbrood virus is also more toxic and lethal on Apis ceranae than it is in Apis mellifera. We don't have that in Apis mellifera. Okay, so remember the dose curve that I was talking about. If you have a virus and you're treating, in this case it's a Nosema treatment. The red line here in the middle is where we started. This is my Sacbrood virus detection out of the IVEDS unit. After 10 days after they treated the bees, we did another test, and we have a significant increase in the number of Sacbrood. So what does that mean that the Nosema fungal treatment is doing to the bee health? Well, the bees are struggling with it. Obviously, their viral load is peaking because the bees are not doing well. Okay, but 16 laters after the treatment, 6 days after the treatment, 6 days after the treatment. Okay, 6 days after the treatment, the viral load drops down. These bees are doing well. Their bee weight, I don't have that on the slide, but their bee weight is doing well. Okay, so I'm going to be quick about this. We've put out books. Charles has put this out. It's documented in 2 books, Integrated Virus Detection System. This detection system is unique. It does not use primers. It does not use reagents. There's no chemicals involved. It is, other than the butanol, which is a condensation part on the particle counter. And Charles is my older brother. I've always played with his toys over the years, and now I'm really having fun playing with them. The other book we have came out a little bit later. It's Identifying Microbes by Mass Spectrometry, Proteomics. I have that in my lab as well, and we run that, and we are able to, we've extended that into looking for gut biome. We look at all of the bacteria in the bee gut. We look for the bee gut bacteria in the bees and get that right. So we don't separate the bee gut. We look for the bacteria that's in there, and it's a real interesting chart. We also look for, I didn't include these charts, but I did look at foul brood, American foul brood and European foul brood. It's there, very low levels. pretty much across the board. It's forensic levels detection. And you'll like this, that now we also look for the environmental microbes, and we see that in the environmental microbes, we chased one beekeeper that was struggling with his bees and they were dying. And we found, we looked at his the environmental microbes, which they were generally collecting from water. And what did we find? We found that the bees were dying. The water hole that they were going to was really contaminated. It was a nasty water hole. All the rest of the bees had pretty much clean water. Wow, stagnant water, probably should stay away from it. But if that's all the bees got, that's what they're going to go for. And this year we put out another book, Microbial Diversity in Honeybees. These are available at CREC Press. Okay, so this one, I included a segment going into looking at coronavirus. And we went to the 120 different sequences for the SARS-CoV-2 that we are all dealing with. And we found them on the honeybees. What? Okay, as far as I've been able to tell, the honeybee cannot host the coronavirus. What? How did I get them all on there? Well, it goes back to Jerry and I at the Army, and the bees are little electromagnets. They're going around picking up all the fragments and everything that's there, and the bees bring them back, and wow, we were able to pick up. It didn't affect the bees. The bees that had no viruses. We ran them through the IVEDs, blank. We ran them through the proteomics at the forensic level. They were picking them up. They were environmental SARS-CoV-2 coronavirus. Wow. We picked them up in the Midwest, and we ran those samples, and they were the highest count that we had for the forensic level. That's pretty interesting. And then we ran them back through, and you know, a couple weeks later, those Midwest, the Illinois samples ended up with the population had their coronavirus outbreak. Is there a tie there? Maybe, but I know that the bees don't carry the coronavirus, but they are a little electromagnets, fly around, and they pick up all sorts of stuff. Interesting, eh, Jerry? So here's my lab. This is what it looks like in the front. These are our concentrators. These are our filters, hollow fiber tangent filters. We run the bees through that. We grind them up, and we make bee smoothies. And we run them through a centrifuge, take the supernatant, run them through these hollow fiber tangent filters, which concentrate, remove all the big things, everything bigger than the virus. In the background, you can see from the picture on the front, there's the IVEDS unit. There's my bank of computers, a bunch of charts to look at it. And there's my prep station over here, airflow, and that's pretty much it. And it takes about 20 minutes for me to prep that. We cut our centrifuge time down to about 20 minutes. We run about 10 samples an hour through this, which is not bad. And then our results come back in about a week, sometimes quicker, sometimes a little bit longer, depending on how long I got to play with the proteomics. So, Jerry, I am done with about 4 minutes to spare, I think. So, any questions? Any comments?
Well, thank you, Dave. And I know that you have that. How many different viruses at the moment does your laboratory screen for?
Well, you know, the easy answer, Jerry, is I screen for all of them. How many have we definitively found? About 30 of them. And that includes that we just isolated and found the Lake Sinai viruses, and we were able to identify that. And I found a couple papers from an author out of Bozeman, and they went TEM and they found that the sizing of the Lake Sinai virus, and I had a big blank in there because I pick it up and I left it as an unknown. And I go, wow, look at this coincidence. I now have a connection for the Lake Sinai virus. I don't have all the variants, but all the variants, the Lake Sinai virus, they're all the same size. They're the family size.
Okay.
Then, anyway,
Okay, so James asks, are viable virus particles and non-viable particles both the same size?
no. So that's an interesting. We looked at capsids that the bees do produce, and that does cause a little bit of a size shift. And we do see that and they shift about 2-3 nanometers. But we will have a peak, and that'll usually end up being a shoulder off of the peak. How many non-viable viruses do I have? Well, we looked at non-viable coronavirus, and they had, I got 0. It was sent from me from BEI Resources, the people that make the sample preps for NIH. And the fragments, I had 700,000 counts on the fragments at a peak. And that's where that verified, that came back through. We looked up where the fragments should be, and that was where they at. So if you heat the virus, their little RNA viruses tend to hold together. If they're not viable by irradiation, they're going to change in their size a little bit. If they're heat shrink, they're going to change in their size. If they are naturally not viable, they will probably remain the same size or very close to it. But it's if I'm following over time and I go from 100 counts to 1,000 counts, I would probably say that those are viable because they're replicating. That's one of the key signs I look for, is the numbers that I got. Okay, I hope that answers the question. Yes, I will see them, and I can by multiple samples I can determine whether they're viable.
Okay, and so he also asked, whether does the electrospray DMA and PCR compare in sensitivity?
How do they compare in sensitivity? I'm 10 to the 2 in my sensitivity. I worked with the NARL lab, the Native American Research Lab at the university, and we were looking at viruses in the thermal pools at Yellowstone. That's basically a pH of, what's the pH of a thermal pool at Yellowstone, Jerry? 2? And what's the temperature?
It depends on the pool, and Dave knows I was a naturalist in Yellowstone for a few years. So north, you can get a pH of 1, and temperatures about are superheated, is on above boiling.
So
most of the pools are cooler and more alkaline.
So we have viruses living in boiling acid. And there's not a lot of them. And we did a TEM, we did a survey on the sizing, and we did a survey on the particle counts from other technologies. And we were right there with the other technologies, and sometimes we were way ahead of them. And I can get down, we can count to, I just did a project for the Department of Reclamation, looking for viruses in wastewater in San Diego. And the challenge that they put out was to be able to find seven individual virions in a liter of water. Okay, so what's my sensitivity? 7 virions. It took me 2 hours, but we did it, and showed the Department of Reclamation and the EPA that the IVIDS technology would pick out 7 virions out of a liter of water. That's pretty low. I can't hear you, Jerry. Sign language, here we come.
Sorry, I turned the microphone off. That's my problem. All right. So along that line, again on the viable and non-viable viruses, the question is, are viable viruses exogenic and non-viable endogenic?
I don't know. That's a good question. Jerry, what do you think?
I'm not guessing on that one. You're the expert.
I don't know.
Maybe Dewey knows. I see Dewey's here. No, he's shaking his head no. All right. Another question. Are you conducting honey analysis to test for various viruses, especially COVID?
Okay, so we have looked at honey. Golly, I wonder how I get associated with honey. There are no coronaviruses intact in honey that I've been able to find. Okay. The genetic markers, yes, we have found traces, but they're at forensic levels. They're very low. I would probably put the Montana Forensic Police Lab to shame at what we can find on the forensic level for coronavirus. The confidence is high. The counts and detection levels are very low. So yeah, Jerry, with your chemical analysis on the bees, you know, we can pick up traces.
Yeah.
So yes, I picked up traces of coronavirus in honey, but it's not there.
Yeah.
There's no coronavirus. If you don't have an intact virus, guess what? It's not infectious. I've never seen genetic material be infectious. Never.
So Dave, I'll add to that question, and that is, you know, the general philosophy of most of the bee books and so on, that honey is rather a hostile environment for most microbes. Do you see any much evidence of any of the microbial, the viruses and other or bacterial things and so on in honey?
The quick answer, yes. Next question. Okay, so yes, we pick it up. I can also tell every plant that the bee has gone to out of the honey. And so we look at that, and we're picking up all sorts of microbes. I pick up tree viruses. I pick up tree microbes. I pick up soil microbes, and very, very interesting details. So there's also a lot of other stuff that we pick up. So are there microbes in the honey? Well, what is the best Manuka honey? If you looked at Manuka honey, why do people buy Manuka honey? Because it's good for your health. Is it good for your health just because it's got sugar in it and plants? It's good for your health because it's got a lot of stuff in it that is really beneficial to the human.
Okay. So let's see, questions on COVID. So there, you know, there's a couple. Let's see. I think I got another. Isn't AFB found in honey?
Isn't AFB, American Foul Brood, found in honey? To be honest, I actually haven't seen it. How's that? I'm looking for it. I have not seen it. I haven't seen European Foul Brood either.
Okay, well, that's good. Of course, that'd be for your proteomics because that's a bacterial.
Right. Well, both of those are the proteomics.
Yeah. Okay. Now, I remember when we did the on that study that we all did with you and your brother, and we published in PLOS One in 2010. What we mentioned in the paper, but most of the readers didn't pick up on, is there, I think it was 900 plant, you know, identifications we made from that thing, and I don't know how many hundred of other things, and we found human diseases and so on. One from on our university, because some of the labs that we're looking at, well, this was the one that always shocked us. Those looking at the sexually transmitted human diseases had active work in the labs, and their filters on the top of the building apparently didn't have biofilters on them, because we were picking up from an observation hive flying out of that same building. We were picking up indication of contact with human STDs.
Okay, so let me add to that. There's been a bunch of work that I've done since then, and that comes back down to, I think their filters were in place. The viruses can go through it, and it is most of the time they use an arduous path for the virus to go through, and they break it up. But what did I say earlier on the honeybees picking up the coronavirus? Oh my! They're not picking up the coronavirus, they're picking up those genetic snippets that fit into the national database out of the sequencing. So we were picking up all of that stuff. That's how I pick up the plants. I pick up the plant viruses. And, you know, you just come back. If I've got an intact virus in the honeybee that comes out to be the intact, that happens to be an almond tree virus, oh my, that's really significant. But yeah, you know, Jerry, that work was incredible. Look at the path you put me down. Thank you.
Well,
you
and I think that brings
way.
up a point I wanted to make here, and that is you initially, historically, you said, both those technologies are available for the U.S. Army, and we use them on some of the colony collapse studies and so on. They actually provided those services. Those have been transferred to you. Initially, you had the IVIS machine, which you've been talking about mostly tonight,
Right.
but you also have a proteomics system now that uses the unique microbial detection software from the U.S. Army, right?
Right.
And so the proteomics looks for peptids. You're talking about snippets or breakdown things. They're looking for peptids that are indicators of. There's whole databases on peptids for almost an unbelievable amount of things. And the essence of the unique software from the Army, that was used in our original study and that you now use, and again, correct me if I'm wrong, is that you look at not just a bee database, but you can look at every single database for every organism out there and screen them, and in rapid time get back the broad spectrum survey. And that's why when we did the thing, people said, well, when you reported in PLOS One, you only looked at bee diseases. Well, that's the focus of that particular paper. We could have written 20 different papers on different things we found.
We still go back to that database because all the other data is there. When the mass spec breaks it up and we record it, all I got to do is add another target that I'm looking for, another plant set, another bacterial set, another fungal set. I just add it to the database, run the software again on that raw file, and boom, I have the data. Yeah. It doesn't decompose as it does in my freezer.
Yeah, and again, I think for our listeners, maybe you're not immersed in this like you are. What you're saying is that the computer has the output of all the peptids it finds, matches it to all the matching things it can find in all the databases and so on. But you may pick up things that are unknown because you don't know where they're coming from. But maybe somewhere down the path, somebody says, "Oh, these belong to this," and you don't have to reanalyze that sample. You got the sample completely analyzed from whatever time, whether it was yesterday or 10 years ago. And so you can retrospectively go back and look for it. That's the nice thing. When we do a lot of the work I was doing when I was originally doing both chemical and biological monitoring with bees, is you get a question about, well, here's a chemical, here's a pesticide showing them. Was this pesticide here before, or was it just because not detected 10 years earlier because the instruments didn't have the detection sensitivity? What you have here with these systems is you've got, uncanny detection sensitivity. And even if you don't know what you find, somewhere down the line, when somebody puts it together, it eliminates the whole need to have the actual sample, because you still have, if you ran the sample through again, it would give you the same answer.
Exactly. So I don't have to run the sample again through the instrument. I just go get the output from the instrument, and I can run that again through the software when I add new sequences to the software, and boom, I get the results, whether that target is there or not.
Okay, so I've got a couple more questions here, and then we'll give people a little break before Dewey comes on. But one of them asks if you see botulism bacillus in honey.
No, I haven't seen botulism in honey.
So the question actually was, how common is it? And I guess not very.
I can pick it up out of my chicken coop. Does that count? So the bees must be selective and they don't go to my chicken coop, right?
Yeah. Well, there's always been some concern over the years, especially for infants, about consumption of honey.
I have not tested every honey around the world, but I have not shown it in our honey samples. We have looked at soil, and we found it in soil.
Yeah, and I will have to admit that much as we'd like to believe that honey is this pure substance that essentially is an adverse environment for a lot of microbes and so on, I have seen a couple of glaring examples that essentially say we can't always trust that. I mean, just like any food product, that's how it's handled and so on. The one that scared the most out of me was when a person who had dog kennels, one of the beekeepers set bee yard nearby, and she noticed the bees were in the watering trough of the dog chemical, or the dog kennel, not kennel. And so then her kids got sick for a couple things, and she got to wondering. Eventually, we ended up looking at some of the honey from those colonies that had the bees that were taking water from the kennels. Turns out we found dog parasites, whether they were viable or not. But we found, under the microscope, you can see the dog parasites in the honey.
Yeah.
That's not honey you want to eat.
Yeah.
So,
Okay, here's an interesting concept for you to consider. How many microbes and how many plants and how many things do I find in honey? A lot? Would that be a fair answer? I usually pick up a lot. What happens if I don't pick up any? Or I pick up a few control items that are there, such as I just pick up cane sugar. What does that tell you?
you going to answer that question?
I could. It tells you that it doesn't have a plant source other than cane. And where did the bees get cane sugar? They're either out in the feed yard picking up sugar out of the feed that the commercial beekeepers are using, or it's not honey at all.
Yeah.
There's a couple other things that we can do to measure that, but honey is pretty diverse, as you were saying. There's a lot of stuff in honey.
Okay, so I got two. I'm going to combine two last questions here from Anthony. So I think you touched upon that. Could you use it to detect adulterated honey? And then the second, and that's probably a yes/no answer, but the second question is, and I think this is the one we should conclude on before we give people a little break and then go to Dewey. What's the takeaway talk for beekeepers as far as what should they be looking for in dealing with Sacbrood? And I would also say, what's the takeaway to them in terms of what a laboratory like you can provide to them? When might they use your services and so on? So let's start with the adulterated honey.
Okay, so the adulterated honey, yes, not conclusive at this point, but yes, I can pick up adulterated honey.
Oh, and I will throw out this one. Can you pick up COVID from humans and surfaces and so on?
I do screen people. Jerry, you were out there. We had one of your chemistry professors come through the lab, and I had spit in a beaker, and then we filtered it a little bit and ran it through the instrument. And sure enough, there is a virus. It's the same virus as my son had that was at a fire camp. And my son called it Camp Crud. And I asked your professor, I said, so how come you have Camp Crud? It picked it right up. So can I pick up coronaviruses? I am screening people. I was at the Western Montana Fair, and we were screening people at the Western Montana Fair. I only had 2 positives out of everybody I tested.
Good. Okay, so we're running out of time. Let's move to the takeaway. What's the takeaway about Sacbrood, and how did they contact you, or if they're interested in follow up of accessing your services?
So right at the very front was my business card. It's 406-777. No, wait, that's too old. 406-369-4214 is my phone number. My email is mrwick@montana.com. Spell out Montana, otherwise it'll go to the state account and I don't exist. The takeaway is, Jerry, the Beekeepers funded my lab. I work for the Beekeepers. I don't work for the university. I don't work for the federal labs. I don't work for the state labs. I work for the Beekeepers. So testing, the Sacbrood virus, monitor it. Over time, it'll tell you what your bee health is doing. If it is increasing, something else is going on. The easiest way to take care of viruses is to give them protein. Marla Spivak and Gloria DeGrande Hoffman and I were all at a conference, independent of each other. We all came up and said, it's kind of obvious to each of us. Each of us said it in our presentations that fat bees are healthy bees. And so we measure the bee weight, thanks to Steve Parks. He told me I better start measuring the bee weight. So I did that. I tied that to Sacbrood virus, and I watched the Sacbrood virus increase. I watched the bee weight drop, and the performance of the bees goes down. It is an easy monitoring tool. My turnaround time is about a week. The cost is two methods. I got a cost of $50 a sample if you tie into a subscription with me, which is $348 a year, whether you send samples or not. And then your samples are guaranteed a rate of $50 a sample. And I can run quite a few samples. I think since we put this in place, I've run nearly 20,000 tests on this instrument. It's pretty robust.
Okay. So if they, so I think, and again, because I know what you do, you've got some of the commercial beekeepers and some groups that are doing studies and so on that routinely send you samples, and you often put that on a subscription service. But if a beekeeper were to walk out and see something really strange with their bees and something's wrong with their hives and so on, they can just pick up the phone, right? Call you and
pick up the phone, call me, email me, text me, any of those combinations. And I'll talk with you. If I'm really busy, I will still talk with you. But basically, it is when I get one sample, I don't know whether that's an increase or a decrease from what the previous sample was. And when I get two samples, I know I've got a bar. And if it's on the way up, it's on the way up. If I get three samples, woo-woo. So every few months, 4 times a year, send me a sample. And then you've got a tracking mechanism that tells you what's going on. If you're more curious than that, run more samples. It is really interesting data. Well, but wait a second. It's really interesting to me. And if I can help you with your bee health, that would be really interesting to you.
Yeah. And what I see from it is, say you wonder whether or not it's worthwhile feeding protein in the fall or so on. You can send you a sample before they can do whatever they're going to do, treating their feed or so on. And then post that, they send another sample. And if they do, it's a way of over time trying out different management strategies and so on and seeing how it affects the overall health.
Absolutely. Absolutely. So an interesting thing about another virus, Jerry, cashmere bee virus, not that I'm interested in viruses, it's pretty benign. Cashmere bee virus is it's just there. And it's not everywhere. When you have mites in cashmere bee virus, it'll kill the mite, it'll kill the bees in about 4 days.
Okay, so one final question, then we need to take a quick break before we put Dewey up. Is bee weight related to the fat body in the bees?
It could be related to just water in the bees, but it is consistent. And I pick it up out of several hives. The beekeepers that I get bees from, when I'm monitoring them over time, their bee weight is very, very consistent. So is it the fat bodies? It could be, but is it the water in the bees? It could be. Is it the size of the bees? Oh, yeah, definitely. So some beekeepers have bigger holes that they brood their bees in. You've seen those combs, the larger combs. Their bees are fatter. But when I compare that bee to another bee a month later and compare it to another sample from the same beekeeper another 2 months down the road, we can show the bees' weight going up and down. And you would think that the lightest bee would be those foragers that come back, their wings are frayed and they're in a honey flow and they're just absolutely beat up. Well, guess what? Those bees weigh the same as the worker bees inside the hive, the same as the nurse bees. How about that?
All right. So I think we should thank you for your talk tonight. Again, I stress that Dave is always very approachable. And if you got a question, call him up, but don't send him samples before you call him and check with him. It's kind of the same way as if you're using the Gastonia lab for pesticides. I really like seeing a lab like Dave's because he gives you chances to look for biological issues in your hives. A lab like Gastonia gives you a chance to look at pesticides and so on. And over the decades I've been doing pollution work and so on, I don't know how many times you think of it as a chemical and it might or might not have been chemical. You think of the disease it may or may not have, but it's not, our eyeballs aren't very good at telling which is which. And so there's a lot of times when the wrong thing is blamed for the problem going on. But if you've got a good chem lab and a good biological lab, you can actually start to understand what's happening. So thank you, Dave. And
let me add just one comment to that, Jerry, is that I also look for nosema and I also look for mites.
Okay. All right. So we're going to take a 5-minute break here. Some people need to maybe run down the hallway. And then we'll put Dewey up and he'll be talking about the Honeybee Health Coalition. So see you back right at the top of the hour.
And thank you. Thank you all very much.
Thanks, Dave. I got one last comment from the questions. Anthony said, thank you so much for answering my questions. This was a very interesting talk. So,
well, great. Sometimes I don't know, Jerry. Oh man, so there I am. So did I stop sharing my screen, Chris?
Yeah, you're not sharing. You're fine.
Okay. Oh yeah, that was me. I wasn't sure if you forgot, and
I did forget.
I didn't think you wanted people to see your email.
Yeah, that's what popped right up.
Chris.
Thank you, Chris.
No problem. I worked with somebody once. I had a job where I had to do training through something like Zoom, and at one point the guy went to his desktop because he was trying to figure out how to log into one of the programs he was showing us, and he had an open text file that had every password and username for all of his stuff open, and he was just scrolling through it leisurely and
screenshot.
Well, my best one, I don't know if Dewey's heard this one, but I was talking to one of the beekeeping groups on the opposite coasts, and they were using a system like we're using here. And anyway, they had all their club members live, and the little boxes up there, it'll dial in, like they ran their club meeting, their business and type of things. And then I was supposed to talk. And so I came in about 15 minutes early just to be sure that I was up and running fine. And I come up, I'm looking at those little windows and pictures. Here's a naked guy sitting in front of his Zoom. and then he got up and walked off. Fortunately, his camera was at the height. You just saw from the waist up and so on. But I thought, well, surely he's going to think that his camera's on. And I didn't have host capabilities and so on, but all of a sudden somebody woke up. He came back, he sat back down in the chair. He had a candy bar that he opened up, continued to eat while I'm sitting naked in front of everybody. So, and then I had to talk. I mean, nothing like throwing them a curveball at you.
Oh yeah.
How many politicians have been tripped up by the open mic, huh?
Yes.
I mean, you don't have to have the full frontal, the open mic sometimes.
Yeah. Well, eventually some of his own club members woke up, and all of a sudden I saw it light up. Hey.
Relative to the bee weight question, we do know queen weight is related to ovarial number. There's a good correlation now. Yeah. How fat body varies is I mean, there isn't the fat, that organ is not very plentiful in any bee. There's not much of it.
I wouldn't think it in the worker bees. So from what you were talking about, Dave, on the drones and the brain, do you think the colony is heavy in Sacbrood? That you were talking about the the viability of sperm, but could there also be an impact on drones and mating?
So Bailey came out and said that high levels of, this is back in 72, and he came back and said that it looks like the Sacbrood virus is causing brain damage to the drones and to the bees, and yes, so yeah, you have trouble mating, That paper goes into one of those two papers that I presented.
Okay.
Goes into Dewey answering your bee weight issue. That the queens, when they are shipped, their bee weight goes down. Their sperm count goes down, and they recover after about 2 weeks in a cage in the hive. Was what their study came out. I think that's just really incredible. And Jerry, I think you sent me a queen. You sent me a bunch of queens that died, and you thought they were all just beat up. I don't know if you remember that or not. Just manhandled and traumatic offense that they died. And I came back with one of them, and I go, no, that one's full of black queen cell virus. It is the highest count I have seen in any bee. And that bee died from black queen cell.
Yep. Well, and many years ago when trichomite first hit and so on, we got some queens that did poorly, and we dissected them. We're counting trichomites, and I noticed the data, and one of the students counted it up, and I said, "Gee, you got what's this? You couldn't count them." She said, "Well," I said, "I don't quite understand. You can see a trachomite, so why can't you count it?" She says, "That queen was so loaded with tracheomites that when I cut it open and so on," she says, "everything just was a pile of tracheomites." She says, she says there were too many to count. Hardly, it wasn't like a few mites in there. So I think it's a good time to transition. We are just 2 minutes after the hour. Hopefully everybody's back. It's my pleasure to introduce Dr. Derrick Dewey Caron. I met Dewey in the 1990s out in Maryland when he was at the University of Delaware. He now is professor emeritus and is living in Oregon. Dewey was has been and is still, I think to this day, very active in EAS, and when he came out west, he joined WAS. But he's been working with the Honeybee Healthy Coalition and has represented WAS at some of those meetings. So I asked Dewey to talk to you about bee health and particularly about what the HBHC is doing. So Dewey, it's your show.
Okay, thank you, Jerry. Thank you for the introduction. For those that are not familiar, I was the president of WAS in 2010. Seems like a long time ago. We hosted it in Salem. Some will remember the Red Lion, where we hosted the WAS that time. And am presently a member at large to the board. All right, the Honeybee Health Coalition has been at one point supported by WAS as one of the beekeeper organizations. And so I'd like to try to give you a little bit of an overview in terms of what the Honeybee Health Coalition is and how your support for WAS has enabled the Honeybee Health Coalition to be able to do some of what it is currently doing. All right, the mission, as we've developed our mission statement, is to collaboratively implement solutions that will help achieve healthy population of honeybees while also supporting healthy populations of native and managed pollinators in the context of productive agricultural systems and thriving ecosystems. So we're trying to do this avoid this division of its honeybees or its native bees, or it's all about pollination and not about all of those other ecological services that honeybees and our native or our non-managed populations of bees, of which there, of course, are a lot more than honeybees, are all related to. We do primarily emphasize the aspect of honeybees because that is where we are able to find some of the support in the funding, but also because of the concern that we have for the number of colonies that do not survive, that seem to be unhealthy. It is a large group. It has grown a bit. It is supported by we like to say all aspects, many stakeholders, one agriculture. So it's everything from The universities such as my own Oregon State University, University of Nebraska, University of Maryland, also by the users of bees, the crop groups, various crop associations, by other NGOs, non-governmental organizations. We have a group that is a foundation, the Levin Foundation out of Ohio. Project APHIS, the big groups, the American Federation of Honey Producers are members. So very proudly, WAS then has a seat at the table. And as Jerry had indicated, since we joined shortly after the founding of the group, I have been that representative for WAS. What we are then is a group that attempts to take with that varying membership, try to supply an equal voice for a consensus-based decision-making process. So that doesn't mean endlessly talking about a topic, but coming to some decision, but not in a sense that that you vote. Now, we have a very varying level of funding from groups. And although I'm continuing to represent WAS and have never been compensated by WAS in any of that I've done, I've funded myself for the various activities. We do have a varying level of member funded as an organization. However, that lack of financial support now from WAS or from the groups that do supply more of the funding versus less of the funding does not have an influence in terms of the role. Each of us all have a part in making and coming to a decision. And it's not just beekeeper group. In other words, all the beekeeper groups being involved, but most of the major ones are involved, but also all of the aspects of our industry. Now we are then facilitated by a group that is called Keystone Policy Center. They're out of the Denver area of Colorado. They are recognized, nationally recognized nonprofit, and they are basically to try to focus a discussion and then work in terms of trying to find some cooperative decisions. Now, our way that we are organized is that we are organized into collective areas of focusing on looking at 3 specific areas and then getting that message out. So we have a group that is the hive management. That group has looked primarily in looking at pests and pathogens. And for whatever reason, we have been one of the groups that has gotten off to a fast start. And that's the group that I belong to as your representative for WAS. We have a forage and nutrition group that is focused on nutrition aspects, but more so than nutrition, looking on the forage part of that, although we are segging a little bit more and looking at nutrition aspect. There is a group that is looking at crops, the crop pest control, and how that impacts healthiness or non-healthiness in terms of our honeybees. So crop pest control and management, and as I indicated, more focused on pesticides. And finally, the group to bring that material to you. This is very well facilitated by Keystone, and I'm also on that aspect as well. So I meet primarily with these groups, but I do indeed have input as a representative for WAS for all of the various aspects of looking at those what we see as 3 key areas. So I've got an illustration of each of those areas. So forage and nutrition, some of the ways that we have are trying to influence the influencers, those that have funding aspects. A lot of the information on forage and forage opportunities comes from the federal level. So input into the Farm Bill, which is done every 5 years, and of course to U.S. Department of Agriculture, but also some of the other ABC agencies of the U.S. Then there is a segment in terms of not only the aspect of pollination, but also how what beekeepers do in terms of pollination, in terms of it being sustainable, supporting not just habitat, but also pesticide stewardship, water quality, soil health, and all of these aspects. And those are important allies for us, because sometimes what will happen relative to a forage being available or not available is related to how much water it may require or soil conditions, et cetera. We have tried to look in terms of trying to help better clarify the nutrition supplement research and development. And at one point, even we're offering Bee Nutrition Challenge, where in fact we then, with our funding that we get from all of the supporters, did fund research projects in the 2018-2019 cycle. And then eventually decided that we are just such a small player in terms of being able to offer funding that perhaps we should focus in helping to support the others. The goal for the Forage and Nutrition ensure honeybees, especially in and around production agriculture, have access to a varied and nutritious diet throughout their life. So to get away from the monocropping and trying to have a better across the board selection of forage, and therefore improving nutrition. Second group, again, these two are I'm not directly supporting those with my effort on behalf of WAS, is the crop pest control. One of the major ways that we've done is support the effort to develop for growers best management practices, or what we call BMPs. So we have helped assist with development of corn, canola, and soybeans in terms of the best management practices, all of them very important, as you recognize, for honeybees, and have now segged on to do some others as well. It is a pollinator-focused continuing education model, so that with the corn growers and the cane growers, the apple growers, all of them, which are members of the coalition, soybean growers, for example, as well, we can have that open dialogue so that they're aware that maybe they do not necessarily need pollination, but there are often still bees that are dependent upon or can make access to the crops that they are representing. We help develop an incident and reporting guide for suspected pesticide exposure events. It's one of several that are available, and this came about basically with information from USDA and EPA saying, what's going on? We're not hearing officially what is happening. You're all telling us that the bees are killed by pesticides, but we're not officially getting that information. So the idea of an incident report was that that information would get through to the relative ABC agencies. And then the grower, of course, and beekeeper fact sheets for pollinator best practices. And then the effort that was developed for the MP3s, Managed Pollinator Protection Programs. Each of the state, or most of the states, developed that effort. Not all of them, but most of them. We were one of the agencies. We were one of the very lead agencies to get all that group together so that they would have discussion. And it had developed very early that the different states were going sort of off in different directions. There was an effort to try to bring that back to ensure that that honeybees, particularly, and bee health was included in the managed pollinator protection program. So basic goal: control crop pests and help to safeguard beekeeping and pollinator health. We have helped develop a bee integrated project, and this is basically taking and developing growers and individuals that use pesticide and pairing them up in their communities, and then have the other walk in the shoes of the opposing group for a period of time. And again, it's all related to this, the best management practice. So we have found funding to help support this. And then the two groups, the grower and the beekeeper, can support, can share that information very directly. So the whole concept is pair beekeepers and producers using the information that we and others have developed in terms of the best management practice. So we're pulling on the information that we've developed on honeybee pests and diseases from that group, for example, the Forage and Nutrition group that I just talked about, for example, and the crop pest management group that I've talked about as well. So using the information that we and others have developed in terms of the best management practices and pollinator protection to try to pair the two together, then give that intimate relationship so that they can see what the other, some of the challenges and some of the successes the other group has for at least for a period of time in their busy lives. So my focus since the beginning has been on the hive management, the third of these various groups. And in that group, we got off to a flying start because we had availability of that wide diversity of not only beekeepers, but those that rely on the bees. And so we developed the resources that many of you have been sharing and using on Varroa. The resources that are available for beekeepers, but also grower groups and growers are using as well. I was a principal author on most of these, or have been at least an author on most of these. I was a principal author for the development of the tools for Varroa management guide. And what this is then the best information, science-based, with best management practice. In other words, idea of looking at integrated pest management approach in terms of how tools that we have, such as the pesticides, such as siting of colonies, the information we have on siting of colonies, drone brood removal, et cetera, to try to integrate that all into one guide. Then, because we recognize some people were reluctant to use some of these other tools, we put together a whole series of videos, short videos, to try to show how this could work. Then, because this is sometimes an oppressive, daunting material to kind of wade through, we came up with a decision support tool, sort of like a dichotomous key, where you supply a small amount of information, 5 questions, and then it will give you an effort of where you might then focus in going back to the tools document of where you might focus on what might be available and what might fit the philosophy that you have, and whether supers are on colonies, those type of questions. And then we made all this information available. So I came up with a way that clubs could take all of the information and give it out, and as many of our members of a coalition as well. We've gone on also with a best management practices simply in hive management. So looking at the product, looking at the mites, looking at the diseases, the foul broods, et cetera. Looking at safety, looking at those aspects of how that may all be put together as in terms of improving or at least helping to sustain better bee health. There's a lot of information in terms of the veterinary feed directive. So we put together a guide in terms of AFB, FB that is available and is being used by both beekeepers and veterinarians in terms of, is this something that is subject to the veterinary food directive, or can I use fluvalinate, for example, for eczema control? Do I have to get a veterinarian prescription? What are the chemicals that are available? We have been able to secure with other agencies some research some funding for development of research and testing of potential new varroicides. As most of you are aware, we are a relatively small market for the commercial companies, and the products we have had available and currently have available have been adapted from some other aspect of animal care. And so this is a unique look at orphan chemicals that chemical companies have come up, and some of their information, it seems like there might be some activity against varroa mites. And so we've had material donated or material made available from them that they don't want to expend the resources to look at, but that we can do. And so it's starting at looking at the very basic screening, whether it has activity against mites, and then a smaller subset that do have activity, looking at some levels, and then bring it to the field and looking at field levels in terms of one of those activities. We have, of course, out there information going all the way back to 2010 that that mites may be developing resistance to amitraz. Some of the indicators that perhaps some populations that have been exposed for a longer time could be developing resistance to amitraz. And so we came up with a case studies of 6 beekeepers across the country, commercial beekeepers, and how they approach the aspect of looking at resistance, particularly the resistance to amitraz and what some of their approaches might be. And now that is moving to obtain a wider collection of samples. We had samples done here in Oregon by Oregon State University. And of course, the USDA through the Baton Rouge lab collected some samples. We'd like to get that expanded and just to get a better read in terms of how widespread that what we believe is developing resistance may be. So our goal with the pest management is to put the best available tools, techniques, and technologies in the hand of beekeeper so they can better manage their hives. And again, what I think is especially valuable is with our group, we have this wide consensus. So it's not just beekeepers deciding or representatives of beekeeping organizations deciding what we should do, but also with the whole total input of this of all the coalition members. On that Varroa sides, it's a wide group. It involves 7 labs in 3 different countries. Has now been going on 3 years. Started initially with 12 plus compounds. And despite in the very difficult year of 2020, there was some field tests that was done, and continuing also with some lab tests as well. So it's going from levels of Varroa vials to larger populations, caging of them, and now it has gone on in terms of hive work as well. So as a result, yes, there are some promising compounds that have been screened and found that might have some activity. And it's being done both in the US and Canada. You can see 14 compounds were screened in 2018 and 18 were screened in 2019. So the number has actually expanded with some additional ones. And now it is in the field testing, which is a longer test period. There's still some major hurdles. It's not that we're going to be able to tell you that there's a new product that's available. Issues that need to be overcome is how to formulate the compounds and develop the right dosage levels. And of course, and then understanding how the compounds are killing mites. You're well aware that we are using compounds and we still don't know how they're killing mites. So part of the lab work is developed to try to find out indeed what is their mode of action, how are they determining it. And then if all of this does result in an effective compound, the huge hurdle is going from the research to the development. So if we can find a way to get it into bees and find that it is being effective in the field test, is then how to move it to market. And that is a real big hurdle, as we found, particularly with some of these chemicals that are not the synthetic materials developed by a chemical. So, in summary, your support through myself as your representative, the Honeybee Health Coalition, collaborative network of private and public sector stakeholders that unpacks complex issues to find solutions towards improving honeybee health. And with that, I might expand also in terms of health of all pollinators. We do have a bit of a focus with monarchs as well, which you understand is I have some issues with their migration patterns, as well as native bees, the non-honeybee pollinators as well. And we do this through engaging stakeholders throughout agriculture, research, government, and conservation groups that help promote multi-factor solutions to honeybee health. So that's a big mouthful. You can find information about the Honeybee Health Coalition on honeybeehealthcoalition.org. If you're looking specifically for Varroa/Varroa, but you find, I think the website is a nice, easily navigatable source to help find your information. Healthy bees, healthy people for a healthy planet. So thank you support for WAS for the support. Initially some monetary support, but now by letting me continue as your representative for the Honeybee Health Coalition. And thank you, Jerry, for allowing me to be able to say a little bit about what the coalition is doing for our members that do not know that.
Well, thank you, Dewey. Actually, I learned a bit about the Honeybee Healthy Coalition myself. And so there's these things once in a while you think, well, I gotta go see what they're doing and so on. And then you get busy with something and you know how that goes.
No.
And I will point out a couple things. WAS has supported, contributed to the HPHC in the last couple of years. WAS is kind of reorganizing and so on. We're working on that issue, and it's a question I'm going to be bringing to the board soon. But you don't have to, you know, with these mini conferences, we're reaching a lot of people that are not necessarily part of WAS. And there's a donation button on the Honeybee Healthy page, and we'll put a link in when we post the recording of this onto our WAS website. And if you think that HBHC is doing a good job, then you as individuals or maybe your companies or so on might want to get involved, be sure to go to the Honeybee Healthy Coalition website and push a button, and they'll be happy, I'm sure, to accept your contribution. And if it's, you know, some company wants to come in as a sponsor or so on, go through Dewey. I assume going through Dewey is probably the easiest way to do it and so on. And I'm sure they'll welcome you with open arms. So,
and if you are a member of WAS and would like to have an issue brought before that, you know, as your director representative, I'd be glad to try to do that as well. So our issues can come from any direction, whether you as an individual, you can support it as an individual, as Jerry has indicated, or through your participation in WAS.
Okay, so we got one question at least already has shown up. Julie Common is more of a comment. She says, "Great work, Dewey. We use your tool in all of my classes. It's so clear. Thanks to you."
Good, very good, excellent. Glad to hear.
So, you know, I assume others may have some questions, but I have a specific question for you. Now they got a little better idea of what kinds of things that HBHC has been looking for. I would certainly agree, you know, the importance of all those aspects. I'm particularly interested in some ways, well, obviously the pest and disease, but the other one is the whole nutritional thing. And, you know, I know you and I have both been hanging around bees for several decades. I'm on my 48th year of working with honeybees. And I see both the backyard beekeepers and the commercial folks, and we do research, done a lot of different types of research with both groups. And if I were to be asked what it is that I think is the number one place where I think we really have fallen down and we really have very little information compared to what we should have, and that's on this whole nutrition issue. And I can tell you the diversity thing really comes in because we've looked in the corn belts and in the canola belts, looking at the neonicotinoids, and we collect the pollen, but we use that pollen to look at the vegetation and the forage that those bees are going for. And even in the most continuous area of corn in the corn belt, where you don't see anything but an asphalt road and the corn rows right there, those bees essentially will find, you know, an amazing amount of research. It's minute compared to what they may need for their nutritional needs, but we have the diversity is still there. I mean, they really work on diversifying what they're collecting. And what we found in the corn belt actually is that the hybrid corn raised for livestock feed or for alcohol production, that field corn has been over the years decreasing in pollen content. It never was very good in the pollen content of the pollen, but it's even worse now. But the bees, in many places, we had to switch in colonies because we couldn't even get bees to fly the area and, eat, whether it's inadvertent picking up of the corn pollen or not. They certainly were not going to the field corn for any pollen. But over in the sweet corn varieties, you would see bees buzzing the sweet corn and harvesting pollen. So this gets into a whole bunch of work we've done where we've seen melon fields and we had colonies under tents and 100-foot long tents, and we've had worked with breeders of new soybean varieties. And there's two issues here. There's a diversity issue, and as I say, in the Corn Belt, they were always trying to find something other than corn, especially in field corn. In the canola, even though 80% of the pollen or more tended to be canola, and they really like canola for both pollen and nectar, they were still looking for other things, around the area. So the bees, given the choice, will actually go out, clearly they want to diversify their diet. But
soybeans, the same story in soybeans. I mean, if you're located near one soybean grower, you can realize their crop. If you're located next to another one and there's nothing there for the bees.
Yeah, and so I've decided our Corn Belt is a green desert.
Really?
I mean,
it really
basically,
is,
I mean, a beekeeper might want to complain of something about the corn, but it's basically no thinking beekeeper should be expecting that to sort of support bees. But the other thing that we ran into is we hear a lot of conservation groups. The whole diversity thing is very important in my mind, but the other issue is if you think of landmass and vegetation, it's the commercial crops. And what we've seen with soybeans is new varieties. One gentleman used a proboscis extension assay system that we provided him. Unfortunately, he passed away recently, but he looks at 14 new varieties of soybeans. Again, they were exploring for higher yields and also whether or not they needed pollinators. And what he came up with of those 14 new cultivars, a lot of them, 10 of them, had no attractiveness at all to bees. They just, they weren't drawn to it. And
that's one of the things we were able to put in the best management practices that as you're testing new varieties, that may be a factor to look at. And often with the breeding programs, that is very far down on their list. How attractive might it be to a pollinator? Because they're just not sometimes aware of pollinators.
And the other issue we found out was particularly with melons and a few other crops, but we've got about a half a dozen major crops now we've looked at. Corn was the same way. I grew up, we pioneered field corn in Billings, Montana. We were one of the first in Montana to raise field corn and so on. So I'm quite familiar with field corn. And we flood irrigated. And I hated to walk into the cornfields when we were flood irrigating if they were in pollen shed, because you just get flocked. Well, we ended up pulling 50 tassels off of 50 different corn stalks on the Corn Belt a few years ago, simply to get enough pollen to make up a minuscule sample for chemical analysis. You had to have 50 tassels just to get enough pollen to analyze. And,
I mean, that all of us have probably a number of us have been through that having to go in a cornfield and bagging the tassels, and oh my God, there's nothing you just come out all cut and beat up. You can't shower that smell off of you at the end of the day.
Yeah, but the thing is that you're talking about a situation where you still got a lot of pollen. What I'm saying is there's no pollen coming off of those things.
On some of the varieties, yes.
Yeah, some of the varieties, I mean, these field varieties are not attracted to the bees. They're having very low yield of pollen, and that pollen that is there is really low in protein content.
Yeah.
And we saw the same damn thing happening. We saw the same thing, I know, in melons. Those tents we had, they were not producing much nectar or pollen at all. 100 feet long tents over 6 rows and so on. And they'd be all yellow, and they weren't bringing back, we were catching individual bees just to get the samples. Now, we grabbed the colonies brought in with by a commercial pollinator. And if those colonies came in, say off of strawberries or something, and they had been or on something that they came in heavy in honey and so on, they lost weight during the time they were on the melon field. If they came in on minimal stores, they had to feed them or they were starving. So on whole fields of melons, bees were basically a net loss. So I got to the point of deciding that you know, yes, we ought to look at the diversity. We need higher quality, diverse, high quality, safe, that is relatively chemical free forage for bees, which we have very little compared to what you used to have. I had the same problem with my university research apiaries. We used to sit on several acres of open land that had knapweed and other things, and the weed guys came in and they got rid of all the weeds and so on.
There
And
you
then
go. Yeah.
they turned, they have one golf course, then we got 2 golf courses. Now we got a bunch of softball fields, and I got a dirt there. I mean, I don't have
Yeah.
enough
Yeah.
forage there to keep things going. So we got all that land use issue. But if I were, you know, the one thing I worry about, maybe you're addressing it for this, is the focus is not, if you look about where could you have the biggest impact in providing quality resource, may not simply be planting the roadsides and between the trees and so on with native vegetation or flowering plant, but I look at what the land's used for, and those monocultures can either be a big problem or they could be turned around to be part of the solution. But we got to get the breeders to figuring out that, hey, they're selected, as you say, for traits that are what they want for marketing or for growth or resistance.
Yeah, for very definitely. And really that nutrition group has been one of the slowest ones to get off the dime and try to get developing programs through our coalition, as well as the people that are interested in nutrition of finding the funding to do so.
Yeah.
OSU, Dr. Segali just got some grant funding, and one of the ways they're going to collect that pollen is the flower constancy of bees is so actually go to the bees and collect the pollen from bee corbiculae and then see what degree it's mono collected and do that work, their analysis that way.
Yeah. You might put me in contact with them, because we got 700 pages of studies that they went to EPA and so on the corn and canola work, and we did every single look. We have several hundred sites in those canola areas and in corn areas where we actually have the pollen profiles from the pollen traps on the front of the hives.
Yeah, and again, because of flower constancy, you can do something like that. Yeah,
and we actually then did some ground truthing on all that. So
yeah.
Anyway, if I had the thing is, I'd start to look back at those big croplands and say, if we can get the seed producers to realize that they're actually they're forgetting about the pollinators, or maybe you're doing everybody a disservice, not only the bees but the crop growers and so on. Because
one of our issues with the coalition has been okay, so I've been a representative right along, and the Federation and the honey producers have had the same person representing their groups right along. It seems out like every year the canola representative is someone new. The apple growers is someone different, and it's often someone that's a fairly new employee. So I wonder
you're
if
going
it is,
back over the
okay,
same
we're
ground.
going to have this event coming up. It's your turn to go. You haven't gone yet, or you're the new kid on the block, and so you're going to go represent our group. And that's, with any group, this is strictly volunteer. None of them are getting paid to do.
Yeah, I understand. That just just taking advantage of you here to say, if I had a thing of saying what I would, if I could start over, where would I put a priority? I'd really be looking at nutrition and understanding that, not only for the supplement thing, but I think we really miscued on
it's
our.
it's not that we haven't looked at it. It's just, I think we've made a lot more progress looking from the beekeeper aspect of the Varroa, and now this amatraz is that's a huge issue for the whole industry, and so that it's not taking time away from the other activities. It's just that they haven't been quite as fast to get off the dime.
Okay, so Anthony says HPAC is a great group, especially helping out hobbyist beekeepers. So they're giving the Zoom presentation to my club in New York in September, and and Anthony says he adds cane sugars to one of these monocultures that some have pollen and some don't, and so on.
Yes.
Yeah,
oh yes.
Yeah. So.
Oh yes.
So. That's all.
One real aside, I supported the PhD of Elton Herbert, and Elton back in the early 70s was the bee nutritionist in the U.S. Department of Agriculture. he died tragically young in his career. And it wasn't really until the Tucson lab picked it up just a few years ago that any nutrition was done within the whole USDA. So it's been neglected by that unit, as well as university units. University people, as you know, have to get publications, and the nutrition route is not like bee breeding, is not a fast track to have a lot of significant papers.
Yeah, and you're not going to make headlines.
Yeah.
Yeah. So I do have a question. It says, if corn is producing such low amounts of pollen, how is corn being pollinated? I'll throw that to you.
Well, of course, it's a wind pollinated plant.
Yeah.
You
You know,
it's got to go
some
down
point-
in all those leaves and get to the silk. Yeah. How is it occurring?
yeah, just kind of like rain gutters down there and so on. But somewhere at the rate we're seeing, I mean, the diminishing yield of pollen, obviously it's still doing the job, but you wonder at some point whether they step off the deep end and get to varieties that aren't.
Well, I think that's the final analysis. If you get these that are not doing that, those varieties will not then have any life at all. So it's sort of a back way of looking in terms of pollination.
It
but one
can't
that
self-eliminate.
will still work. Right.
Yeah.
Exactly.
So let's see. Okay, somebody says here, if we concentrate on bee nutrition, we may find that healthier bees will have more defenses against mites and so on. I think that's exactly what we heard from Dave.
Yeah. Healthy bees, healthy
Yeah.
planet.
Well,
Yep.
we found the same thing. We did a study for the Crop Dust Research Consortium. We did a couple years with them. And of course, there's this whole planting dust problem with neonics. And most of the groups really during the studies they did, and they did it for 3 years, and there were some in Canada and some in both parts and several parts of the US, didn't actually see these occasional reported bee kills, except we did. But we picked it up in Nebraska, not during the planting, but during this right after a snowstorm before they started planting. And I have a pretty good idea where it's coming from, and it isn't crop dust. I think.
It isn't crop dust.
It isn't crop dust, but anyway, but then we got to wondering, we took a pretty severe hit. We had 30 some fields and we had bees on, and 3 of them, I think it was, that we had some severe bee mortality out in front of the hive right at that snowstorm. And it took another week or 2, and then they did go into the planting, and then we had 8 weeks of study. And by the end of the 8 weeks of study, we couldn't tell which of those colonies had been actually hit by a kill. And we did the chemical analysis, it was neonics again, but it wasn't from the dust.
Right. Not from the
but by the end of about 10 weeks, you couldn't tell the difference between the ones that had a huge kill and those that didn't. But we had enough experience in the Corn Belt that we figured the only way we were going to get these things through 10 weeks and make sure we have viable colonies for collecting samples for us of pollen and that we could take the bees from is that we actually supplement fed them all the way through, just as a safeguard to make sure that we did everything we do to keep them healthy. And out the other end, they look so well that we then got the Crop Dust Research Group to allow us to, in another year of a long-term, what happens if you nuke a colony with neonics and then you feed it versus colonies that aren't nuked and the controls were both, we had nuke colony, we actually did it here in Missoula because we don't have much neonic use around here. So we're kind of neonic free. So then we spiked the pollen, we dusted bees with neonics and an implosion chamber. We're using a whole size colony, but we pull a couple frames out and then put them back in.
Yeah.
And then, so we had controls. Half the controls did not have any exposure to the pesticide. Half the controls did, half the controls were fed, half the controls weren't. And of course, they didn't have any pesticide. And the treatment was pesticides, half of which were fed and half of which weren't. End of the summer, the data showed they were all surviving and there was no significant difference, but we did induce the same type of kill we saw in Nebraska. I mean, we used traps and we literally had to keep going up and concentrate till we got to the level of kill we saw. So it was a pretty severe kill, so there was several thousand bees out in front. And then we took them through a whole winter. Crop dust folks thought, well, we did 6 months. What do you worry about? No, we want to go through a Montana winter. That separated it out. The next spring, though, the interesting result was, if you were fed either both the colonies that were nuked with the neonics and the control colonies more or less did the same through the winter and had pretty good survival. But if you weren't fed, both the controls and the ones that got hit by the nukes, you were nuked. But the nuked ones there were pretty well wiped out. So we have our own study that suggested that, proactive feeding of supplemental feeding right after pesticide incident might be a good strategy, because it may not affect the outcome, say, by that fall, but if you go through a winter, by the next spring, you're likely to see a big difference. And so
and proactive feeding during pollination, and that could relate to pesticides, could relate to the fungicide connection with sterols. It could relate to a number of things. But again, that proactive management of colonies versus simply putting them there and not, not looking, they're strapped. So I'm not going to go into those colonies, I'm not going to do anything, et cetera.
Yeah. So there are some, so I'm leaning more and more, and as Dale Hill, who wrote the chapter on the hive and the honeybee from 2015, he teaches our courses. He is an animal, was an animal nutritionist and worked with other animals, but then transitioned in his retirement and went to work with data and made the supplement up. And Dale said the same thing you said, that the U.S. nutritional research is really scant, and most of the work being done on that is in other places like overseas in Europe and so on. If you're really looking for that, so.
Yep.
Well, let's see. Oh, I got a couple more questions, I think. Thought I saw another number come, but I don't see another question here. Well, Dewey, I really appreciate you giving us an overview of the coalition and what it's doing. And I from the comments we're getting, it sounds like people enjoyed it, found it informative and enjoyable. And hopefully some will step up to the plate and help you, the coalition out, either individually or maybe with their own companies and groups that they know of or associations. It's been a good evening talking about bee health. And I think we covered, down from the specific analysis to some of the strategies and so on that can be used to improve bee health. So I'm glad, if nothing else, I'll be very glad to put this into, okay. Oh, I think I'm going to be glad to see the recordings of this added to the WAS website and so on, because I think you got a great presentation there, and it really encapsulates the information in one place. So I thank you a lot, Dewey, for taking, I know you had a double hit today on talk, so.
Yeah, I flew from Ohio to Montana in that half hour. So.
And Linda has a question for Anthony about the group in New York that's going to get a Zoom session. I think she wants to connect up and figure out, I'm guessing she wants to know if she can get in on that one. So.
Yeah, SABA, it's called SABA, S-A-B-A, Southern Adirondacks Beekeepers Association. SABA, S-A-B-A.
Okay.
They have a lovely website.
All right. Well, Dave, I see you're back. Dave and Dewey, I'm glad to see you tonight, and I enjoyed the night, and I'm sure from the comments we're seeing that others did too. Hold on and see if anything.
Good job, Dewey.
Oh, thank you. Thank you. Thank you, Dave.
So with that, I think we'll let everybody say good night to everybody. We'll see you next month.
Thank you, Jerry. Thank you, Chris.
Thanks.
Chris, yeah.
All right. Good night.
Good night.
All right, Chris, you got to wrap up.
Yep. I'll give it a couple minutes and then I will shut it down, make sure the recording worked, and get that sent off to you.
Okay. Maybe I got the last night for some reason. Maybe I figured out the closed captioning didn't work, but I think I need to be coast on that for that to toggle. But we can play with that before next month. But the one I did for the Seattle folks last night, I read in the fine print that it said something about if you want to keep these closed captioning going with them, send your recording to the cloud. So that's what I did last night. I'll have to run a test here and see whether or not it makes any difference whether the cloud you save to the cloud or save to the computer in terms of whether it saves those closed captionings. But I got back, and it said